Greetings and welcome back to the Old Ways Rising Farm YouTube channel, where we're going to do a bit of a chalk talk today. This will be an addendum to the animal and plant breeding and genetics video series, and it's in response to questions that I've gotten. One of the common questions that I get is, in some form or another, if you can pure breed an animal for everything else, why can't you pure breed it for a healthy immune system? That doesn't make any sense. And I've tried to explain this a number of times, and some people get really incredulous when I try to explain it because it is so different from any of the other systems that you're going to directly interact with. So I wanted to take a minute and do a slightly longer explanation of how the immune system functions and how that relates to the genetic potential of an organism. Okay, what is the interface of genetics and immunity? To do that, I have to start by explaining immunity. How does your immune system function? Where do white cells come from, and how do they work? Okay, this is going to, even though this is going to be a slightly long-winded explanation of a single aspect of breeding genetics, it's still extraordinarily oversimplified because the immune system is one of the most complex and complicated systems in any organism, okay? So I am oversimplifying, but it's still going to take me a minute to get it all out. But at the end of this, I hope that you'll understand why, no, you cannot pure breed for a healthy immune system. And as you pure breed after so many generations, you will collapse the immune system, okay? And I'm not going to talk specifically about one critter or another. Everything that I'm discussing in this video applies broadly to all vertebrates. Plants don't do the same thing, but it applies broadly to all vertebrates. Let's dig into the information. So, why can't I pure breed for a great immune system? Now, I'm not going to go over all of the details of the breeding patterns, breeding definitions, genetic definitions in this video. If this is the first one that you've come to, go and hop back to the beginning of the breeding and genetics series. There's a link at the top of the screen if you want to do that, and then cycle to this one at the end. Now, where I do want to jump into some terms are terms related to the life and death of cells and the immune system. The first thing we need doesn't directly, it's not a direct immunology term, but you need to understand it before we can talk about immunology, and that's apoptosis. Apoptosis is basically a self-destruct sequence that all of your cells have built in. It is a programmed series of events that starts with the cell self-digesting its DNA, and then collapsing protein production, and then the cell is dead. It ceases to exist, and it is torn apart and digested by the other white blood cells. Okay, this is a very, very important process. It is important first in fetal development. So when you're a teeny tiny little critter in utero, all of your fingers and toes are webbed. And then a little bit before birth, as the fetus matures, that webbing dissolves. And that is an apoptotic process. All of the cells that make up the webbings undergo apoptosis and are removed and destroyed. Okay, but the cells that make up your finger aren't. There is a small number of children that are born with webbed fingers and toes. Okay, because the apoptosis is incomplete. Apoptosis is critical for preventing cancers. So all of your cells in the cell division process have a series of checks that they undergo before the next round of division takes place. And if the cell fails any one of those checks, apoptosis kicks in. So apoptosis has to be turned off before a cell line can truly become an immortalized cancer cell line. And it's important in immunity. So you'll see where that comes in here in a few minutes. So apoptosis is programmed cell death. Now, immune cells. We're talking about the white blood cells. All of these are initially, with an asterisk, we'll come back to that asterisk here in a few minutes, are initially produced in the bone marrow. Okay, so your bone marrow contains stem cells which divide in, and some divide and produce red blood cells, some divide and produce white blood cells. We're not going to talk about the whole diversity of cells that are produced by those lines. We're just going to talk about the ones which kill diseases. So those are the T cells, natural killer cells, macrophages, and beta cells, B cells. So T cells, these are the ones that are impacted and killed by HIV/AIDS. Okay? T cells do two things. First, they help B cells activate. We'll talk about that later. And second, they recognize a cell which is infected by a pathogen through labeling with an antibody. Okay, so when a cell is labeled with an antibody, that's a kick me sign. Okay, we'll get down to antibodies here more in detail later. But if a cell is labeled with an antibody, it's a kick me sign, and then the T cell comes over and it kicks it. So it does those two things. So first, B cell activation. Okay, and then when it recognizes a cell with a kick me sign, it comes over and it comes to that cell and it goes, knock knock, anybody home? And the cell says yep, and then it says, here's a ligand which will trigger apoptosis. See you later. Okay, so it's kind of a sniper. It's kind of your like scout sniper of your body's army against disease, right? It sneaks in. Sneaks in, like, kind of stealthy, counterintelligence-wise, and spots, ooh, you're no good. Why don't you do apoptosis? Okay? And takes out infected cells that way. So we trigger apoptosis. Okay? So that's kind of like your counterintelligence force and your sniper force right there. Now, natural killer cells. These guys also are floating around your blood, and when they recognize a cell that is labeled with antibodies, they blow up. And when they blow up, they release hydrogen peroxide and a lot of free radicals. And they just literally burn the cell to death with peroxide and free radicals. Okay? So these are your suicide bombers of your body's army against disease. Okay? So these come in and they kill infected cells with oxidative stress. Okay? And the natural killers are most of the reason that you feel bad when you have a cold or a flu, right? So the burning in the throat, the burning in the lungs, right? All of that inflammation. That's collateral damage from these little suicide bombers going in, blowing up, and just releasing a flood of hydrogen peroxide. Yes, they kill the infected cell, but they also kill a lot of cells in the neighborhood. Okay? And when you get something like SARS-CoV-2 or like a super flu, what happens is that the reason you die from those diseases isn't really because the virus kills you. You kill yourself by these guys being just so hyperactivated that they're blowing up all over the place and you're just destroying your own mucous membranes from the inside out. Okay? So once your body becomes vaccinated or exposed and has survived it once, and then the next time it's exposed, that doesn't happen. Right? So that's the process by which new pathogens, like a SARS virus or like a flu virus, come in and cause the death and destruction that they do. And that's why vaccines are so important, because we can train these guys to act appropriately and not just all blow up at once. Right? That's what we're doing. Now, third kind of killer cell is the macrophage cell. Macro means big, phage means eat. These are literally the big eaters. These are Pac-Man cells that come in, engulf and swallow a antibody labeled pathogen or cell infected with pathogens, and then they digest it. The macrophage dies in the digestive process, but so does the cell that it engulfs. Okay, so these are your Pac-Man cells. I'm just going to label these big eaters. Okay, it's a literal translation of the Latin for this cell type. They come, they Pac-Man, they eat, they digest. They die in the process, but they digest what they ate. These are also the cells which are responsible for cleaning your lungs, because they can emerge from the blood vessels in the mucous membranes and go swim around the fluids on the surface of the small airways. So, if you get dust or debris or smoke or something like that in your lungs, these guys engulf the particles which are contaminating inside of your lungs, exit the lungs, and then digest it in the bloodstream, where their guts as well as the digested dust particle can be dealt with by your body's other filter systems. Okay? So they're very important for lung health. If you get something like silicosis, where you have like little silica particles or ground glass, like glass dust in your lungs or something like that, these guys come in and try to clean it, and it pierces the cell, kills the cell before it can engulf and remove it from your lungs. And that's why those types of dust can cause such catastrophic damage to lung tissue over time. And that brings us to B cells. B cells' job, these are really kind of, if we're comparing these cells to an army, this is the CIA of your body. Okay? These are the intelligence officers that are going out, recognizing threats, and then labeling that threat so the rest of like your scout snipers and your suicide bombers and your big eaters can come in and deal with it. Okay? So your B cells don't actively kill an infected cell or destroy an infectious particle. They produce the antibodies which label it. Okay? So B cells recognize, activate, divide, and then produce antibodies. Okay? So I'll write that down. Detect, activate, divide, make antibodies. Okay, writing sideways. There we go. So this process is rather complicated. I'm not going to draw the whole thing, but you start with a B cell that is being produced again in the bone marrow, right? If a B cell is activated in the bone marrow, it itself undergoes apoptosis. Okay? That's because your B cells need to go out and detect all threats. So some B cell is capable of detecting anything, including your own tissue. If it detects your own tissue and activates, and then persists, that is a common source of autoimmunity. So inappropriate B cell activation is responsible for lymphomas, it's responsible for some leukemias, it's responsible for some forms of lupus, some forms of like autoimmune-related irritable bowel type conditions, it's responsible for MS, it's responsible for some forms of autoimmune-related diabetes. It's responsible for a lot of problems if these little buggers activate while they're still in your bone marrow. So they have to get kicked out of the population. That again happens through apoptosis. But if a B cell does not activate in the bone marrow, therefore it's not sensitive to your own tissues and fluids, it's released into the bloodstream as a young, naive B cell. Okay? Now that B cell is making an antibody with the capability of detecting something, and the more somethings that your total population of B cells is able to detect, the better your immune system functions. It depends on diversity. That's where we're going to cycle back to you can't pure breed for it. Okay? You can pure breed for one type of immune system, but that will only be able to detect a teeny tiny percentage of the challenges that your body needs to face as you go through life. Your immunity depends on B cell diversity, period, point blank, end of story. no further discussion can take place. A population of B cells lacking diversity results in a dead patient. Period. Point blank. End of story. Okay? So what are these antibodies? Well, they're these Y-shaped molecules. It's a protein. So the bottom, what I'm drawing here is an IgG antibody. The bottom is tethered to the B cell. So let me draw just kind of, here's the membrane, and this is a B cell. Okay? So there are 4 components to this antibody protein. You have 2 long ones, which are tethered in the middle. Those are called the heavy chains. And then you have 2 short ones tethered to the outside of the heavy chain, which are called the light chain. Okay? Both have, this is a mirror image of itself. So both heavy chains are the same, both light chains are the same. Now, the spelling of the code for the protein in this outside end, right there, is different for every naive B cell. Okay? And the spelling for the code for that protein will lead to different amino acids being incorporated in that. And it will, the different amino acids incorporated into the end of that will allow it to stick to different things. So the active site is the sticky end, and it sticks to different types of protein. Okay? Since everyone is spelled a little differently, everyone gets a slightly different order of amino acids and is differently sticky to different things. That's why your immune system can recognize so many different items that are foreign to your body. Again, it can make these that recognize your own body, but they die by apoptosis before being released. Okay? So our active site is the sticky end. So here's our B cell, our naive B cell, with this protein attached to it. And now this comes, and let's say we stick to something ugly here. We'll give this a little frowny face bugger. Okay? So now we have our naive B cell with its antibody stuck to a bad guy. A whole series of complicated steps take place once this happens that lead to the B cell being activated. So first, this B cell is going to recognize that its antibody is stuck to something, so it's going to suck it in, digest it, cut off a little piece, and then spit it back out, but still attached to the cell membrane. And it's going to carry around this little piece of bad guy like a trophy. So step 1 in B cell activation is it collects a war trophy. Okay? Now, a naive B cell with a war trophy is going to come and it's going to be recognized by a T cell. The T cell is going to come. The T cell is kind of like the scout sniper, but it's also kind of the general of the army. So the T cell is going to recognize that this B cell has a nasty, ugly bad guy attached, and they're going to come and they're going to huddle up. The T cell is going to give the B cell a chemical pep talk. That will initiate the rest of the activation cascade of reactions. Okay? At the end of the pep talk and the full activation of the B cell, it starts to divide and produce more B cells. And it's not going to divide and produce just one type of B cell. It's going to produce some which walk around with replicas of this trophy, basically, in a complicated way, which will then activate any other cells in your body which are capable of even coming close to sticking to this bad guy. Those are your memory cells. They are long-lived and they last for a long period of time. Again, when you take a vaccine, one, you're training the macrophages not to blow up too darn fast, but also you're producing a population of memory B cells through the activation of the naive B cells that can interact with that vaccine particle. And they will then persist for a very long period of time and give you lasting immunity. But then you also produce a small population of memory cells, which can activate other B cells and retain that information for future infections. You also produce a lot of shorter-lived B cells, which have no job other than spitting out this antibody as fast and furiously as they are capable of doing. Okay? They will live for quite a while, but not forever. So this is why you have waning immunity. And this is why things like coronaviruses in particular, you get a lot of short-term active spit out the antibodies B cells, but you don't get a lot of memory B cells, which is why you keep getting the cold over and over and over and over again, because you don't have the multi-decade lasting immunity of a large population of memory cells. You just get the short-lived spit out antibodies and attack it now cells. So the spit out antibody and attack it now cells, you get, each cell will produce one type of antibody. You can have more IgGs floating around the blood now. You get IgAs, which are 2 antibodies tethered together, and you get IgMs, which are 5 tethered together. Okay? So those are floating around the bloodstream, sticking to the bad guys. And part 1 of this is once they stick to a bad guy, they become a label. So now your T's, natural killers, and macrophages will come, recognize that they now have, this is a particle with a kick me sign on it, and they'll eat it and destroy it. And 2, these ones that are polymers can stick to 2 bad guys at once, glue them together, and that can function to inactivate their ability to infect other cells, especially with viruses. Okay, so some viruses are just, their whole outer surface is coated with proteins that are capable of attacking the cell. Others only have a small number of those proteins, which are capable of attacking a cell, right? So if you gum up, if these antibodies are sticking to the antigens that allow them to attack a cell, well, now that's just a physical blockage and it can't attack the cell anymore. And then these, right, you can, because you have these guys that are polymers, right, maybe you have a virus here, and a virus here, and then those get more antibodies on them, and then they stick to more viruses. So you can end up getting the viruses consolidated into a big old clump, which is easy for a macrophage to eat. So instead of 1 macrophage, 1 virus, now maybe you have 1 macrophage taking out 100 or 1,000 viruses because they've clumped up together. Okay? So these guys are particularly important with the rabies vaccine. Stimulates a large amount of IgM production and just balls up all the rabies viruses before they can get into the nerve cells. Right? So the this process produces those tools that your body has to fight the infection. Now, here's where we circle back to genetics. You have a dozen genes in your body related to antibody production. Only a dozen. You have 6 genes for light chain and 6 genes for heavy chain. Okay? In the immune system, in the bone marrow, when the precursors for your immune cells are dividing and producing immune cells, they will at random, so you have the stem cell, and it's about to produce a white blood cell. Okay? As it goes through that process of mitosis, 1 light chain gene will be selected at random. The rest are turned off. Okay? 1 heavy chain gene will be selected at random. The rest are turned off. Okay? So you go through, and each B cell is a random combination, 1 of the light chains and 1 of the heavy chains. Okay? That's your first step in producing this huge diversity of recognition capabilities. Okay? And this is the core of why your immune system collapses with inbreeding. Because as your genome becomes more and more homozygous, so does the population of genes for your immune cells. So you have 12 genes. If all 12 are distinct, that's your maximum immune system capability. Now, there's going to be more than 12 genes in a population. There's 12 genes in you, but in the whole human population, there's more than just 12 genes, right? So one of the things that's interesting about human history is that, the African, currently there's only one race of human beings alive, and we are all the African race. Okay, you had the European race was Neanderthal, and then the Asian continental races were Denisovans and Homo erectus. But only the African race persists into modern time, but we did crossbreed with our ancestors. So we have ancestral African genes for antibodies. We have Neanderthal's genes for antibodies. We have Homo erectus' genes for antibodies. And we have Denisovan genes for antibodies. Okay? Floating around the human genome. So there's a lot to work with at the population level, but you only have 12. As the anatomically modern African subspecies of human contacted Neanderthal, they failed to colonize Europe several times until after they interbred with Neanderthal. And when we studied the ancient DNA, one of the things that's really interesting is that a lot of the European population genome for antibody production is Neanderthal in origin. So there was something about the diseases of Europe that required an input of Neanderthal immune DNA in order to tolerate the disease environment of Europe. Okay? And now that's spread throughout the whole human population. It's not just Europeans that have it. So there is some ability to breed for a more adapted immune system, but you cannot inbreed to the point that you render the that suite of genes for antibody production to be homozygous. Okay? You need maximum diversity, the maximum potential diversity, right? Maybe this gene doesn't do anything in our climate, so we need to get rid of that and replace it with this other gene. That can be a thing, right? You see this in other populations too, not just humans, right? So there are diseases that afflict domestic sheep that just absolutely tear apart Rocky Mountain bighorns. Okay? So there is a gene floating around the domestic sheep population that is not floating around the Rocky Mountain bighorn population for resistance to a disease that afflicts domestic sheep. Okay? So outcrossing to gain function is a thing that can happen. Okay? But if you are running an inbreeding program, well, maybe the parents of your program have 12 genes, but after a couple generations, maybe you have a couple duplicates in the short chain and a couple duplicates in the long chain. Right now, maybe you only have a maximum of 10 to work with. That's a compromised immune system, but it can probably survive it because of the next mechanism I'm going to talk about. But now a couple generations more, and now maybe you only have, everything's duplicated at least once, and you only have 3 options to choose from in light chain and 3 options to choose from in heavy chain. Now you only have half the capability to recognize new disease, and you're going to start to have immune system collapse. Okay? You can tolerate some, because moving away from just what's in the DNA to what's in the B cell, this active site region of DNA, after one is selected and the rest are, the other 5 are turned off, okay? After that takes place and is done, you now have a second event where the DNA in the active site region is actively scrambled, okay? You're not going to gain a new amino acid. You only have the amino acids to work with that were in the gene that coded for the original step. But those amino acids can be scrambled a little bit backwards and forwards at random. And then this happens again when an activated B cell divides to make its antibody pumping cells. That scramble event happens a second time. Okay? So those cells will be substantially similar to the original B cell, but just a little bit different. So over the course of the disease, there's an evolutionary process in the secondary division of activated B cells so that the recognition gets better and better and better and stronger and stronger and stronger as the disease progresses. Okay? Because those daughter cells have an activation process of their own. Okay, so if this second division makes one which is similar but can't really stick well, it's not going to be activated much. But when it makes one that sticks even better, that will be activated a lot, and then it divides and does the same thing. So there's this culling process over the course of an infection where the activated cells become better and better and better and better and better at recognizing this specific pathogen that's attacking the body. Okay? And that happens once for the production of the naive B cell, but then many, many times in the daughter cell populations over the course of an infection. Okay? So you do have some ability to produce random variability even from a slightly constricted antibody genome. But once it gets too constricted, you start to have just not enough to work with. This extra little bit of diversity can't keep up with the fact that you just don't have the tools in the toolbox to begin with. And that is why you cannot pure breed for a great immune system. As a little addendum to this, I do want to mention that I fully recognize that what I'm saying here is oversimplified to the breaking point in a lot of ways. B-cell activation by itself is worthy of a full semester length 400 level college course. And I'm trying to cover that and a half a dozen other topics in a little half-hour video clip for farmers that are trying to breed some chickens. So yes, I am oversimplifying a lot of things here to the breaking point. I will put some documentation and some articles if you want to get into like the really gory details in the description box below. One of the places where I am absolutely oversimplifying is like number of genes. In modern research, they're really talking not so much about genes as variable coding regions because there's so much recombination of the DNA when B cells are produced. Also, in different species, there's different numbers of genes, but there's a finite number of DNA that leads to a huge amount of complexity. But when you reduce the starting complexity of that DNA, you lose permutations at the end of the story, right? That's the key take home. I'm trying to nutshell it as best I can, but if there's any true immunologists out there, you're, I'm sure, cringing here. They're like, there's detail. Yeah, I know there's detail. The crib notes version. So I hope it helps. I hope that you found that useful and that it explains a little bit why this particular system of the human body, or any vertebrate, as I said at the beginning, is so different from all of the rest of the things that we breed, and or all the rest of the traits that we interact with when we're breeding a population towards some specific agricultural goal. I hope it helps. I just hope it helps. It is complicated. I also hope that you're enjoying the channel, and I hope that this mixture of the agricultural and the making helps you become more self-sufficient and more qualified and capable as a doer of things and grower of food, just in general. And if that's the case, this is an unsponsored channel, and we are dependent on our patrons on Patreon for support for the work that we do here. I am so grateful for the folks that are already over there and would love to invite you to come join. There's a lot of rewards in the Patreon digital store. Has reams of documentation of historic skill sets and plans and patterns, and more of all of that is coming. Each of those documents is available for sale by download if you want to just buy one. But I would much rather build a community, so all of the paid tiers, even the $1 a month tier, gets full free access and unlimited access to the entire repository, which is growing all the time. So if you would like to come join us, I would love to see you over on the Patreon page. But either way, I'm glad that you're here. I hope that you will continue to watch our videos, and we'll see you next time at Old Ways Rising Farm.